By way of introduction, my name is Nick Seaver. I'm a board member of the Med13L Foundation and helping to drive forward the Cure Med13L initiative. I am joined today by Dr. Wendy Chung, who is the Director of Clinical Research at Safari, Dr. Reza Asadolahi from the Institute of Medical Genetics at the University of Zurich, and Dr. Randy Stritch, professor from the Department of Molecular Biology at Rowan University. First of all, thank you to all the researchers for your terrific presentations. I'd also like to thank the Simons Foundation for pulling this all together, and in particular, Erica Jones, who has been coordinating lots of detailed logistics to make this work. And then, of course, thank you to the families for taking time out on a weekend for this important session, and a Saturday that, at least in the Northeast, is a quite beautiful Saturday. So thank you. So I think we have most people now have come over. For everyone listening in, you should be able to see and hear me on your screen. I won't be able to see or hear you. We welcome you to ask questions in the Q&A box. Please remember to avoid including personal health information in your questions for your own privacy. We apologize in advance if we do not get to your questions. We'll try to cover as many as possible. And with all that said, let's get started. Let's begin with a question from the audience. If we are based in Europe, how can we send samples? What is the procedure to obtain a sample? Wendy, might that be for you?
Yep, yeah. I think that's probably for me. So although if Reza has another answer for anyone that might be able to get a sample to Zurich, feel free to chime in after mine. But so as I had said in my talk, we are collecting samples specifically to be able to make these induced pluripotential stem cells. I will say COVID has wreaked havoc in the world in so many ways, and this is one of them. To be able to make the induced pluripotential stem cells, we have to receive the samples very quickly while the cells are still alive. And we have been having trouble getting things through customs internationally, large part because, like I said, people are very skittish about COVID, and they've been having extra scrutiny in terms of anything that looks like a biological sample, and clearly a blood sample does. So for right now, especially, we're not doing international samples to be able to come in. I hope we'll be able to make some adjustments. We do have this set up, though, in the United States. So we'll be making announcements as we're able to do and support other things. And it may be that we do this with, I think of them as sort of European stops. So some other places, other laboratories, other universities in Europe who can more quickly get the samples and process them. So stay tuned. But Reza, I don't know if you have another better option. Feel free.
Yeah, thank you. Basically, we have the facility at the Institute of Medical Genetics at the University of Zurich to receive fibroblast cells, for example, and keep them into the liquid nitrogen where they should be properly stored, and then also then find a way for making the stem cells out of them. So if any patient is interested to contribute to the research, they can talk with their physicians, preferably academic physicians, and we can get in contact and organize that. I mean, normally we get samples from academic physicians in Europe for sometimes RNA studies or other studies, fibroblast samples, skin cells, usually. So this is possible. We can handle that, and in future we could also see how we can interact with Simons Foundation and share the samples. But if at the moment US is not possible, we could organize that in Zurich. But I need to be in contact with the academic physician if possible.
Thank you. We have questions now on CRISPR. And I think I'd like to begin by asking the researchers to tell us a little bit more about CRISPR, as well as newer systems like prime editing, what they are, how they can support non-clinical research, and then how can they possibly even be used for part of a clinical therapy? And specifically for Randy, a member of the audience wanted to learn a little bit more about the ICAP that was used on the cells. Were these just lab tests, or were they in vivo tests? Was one question. And what changes would you expect to see if this was used in the clinic in someone with MED13L? What kinds of improvements might this possibly lead to?
Yeah, those are all, yeah,
That's a lot. So
that's a lot, but those are all really good questions. And that's something that we're all struggling with. So CRISPR, to get to the very beginning, CRISPR, and there's a lot of derivatives coming off CRISPR now. The original idea of being able to go in and make genetic changes, editing as it's called, the genome, has been around for a little while, but the number of changes that's happened to CRISPR, including this ICAP that I talked about in my presentation, mostly that those have done is increase the efficiency of CRISPR and given you a little bit more options on what you can do to the genome and how quickly you can do it. So in our case, we've been able to replace or to correct the MED13L mutation, and this was done in a cell line. Okay, this wasn't done in patients. This was done in a cell line. And it was very efficient to do that. And so there's new generations of CRISPR type technologies being developed constantly. And so trying to keep, it's a moving target to some extent. What CRISPR can do is it's early days for using CRISPR in the clinic. It's been looked at for some single gene mutations in people, so where you can easily take cells out and then put them back in. Liver, for example, they've been able to do that. However, brain is neurons are a little bit more difficult proposition to do because it's hard to get in and introduce the CRISPR machinery into neurons. And it's difficult you can't take them out and put them back in like you can other cell types. So right now it's still a waiting game, and it's still not quite clear exactly how CRISPR can be used, especially in early embryology. That would be that type of correction is difficult. As far as the role of the mitochondria and being able to do those type of things, what we would expect if we could change it, what we would expect would be a reversal of that fragmented phenotype I showed, and hopefully the restoration of mitochondrial function. It's clear from a lot of different studies that mitochondrial defects are associated with a lot of neurological problems. I mean, Alzheimer's, those type of diseases are associated with mitochondrial defects, as well as developmental problems. There's several syndromes out there that would be like that. So what we would expect would be a gradual return of mitochondrial function. Now, the thing about mitochondrial DNA and being able to return that in a neuron is that the mitochondrial DNA replication doesn't behave like nuclear DNA replication. It doesn't happen once per cell cycle. So mitochondria can reestablish themselves if they're given the opportunity to. So different types of therapeutics that would stop, say, cyclin C action on that fission machinery, that may allow the mitochondrial DNA to recover and then allow some normal neuronal function at that point.
Thank you. And how much do we understand at this point about the connection between mitochondrial health and function and the functioning of the central nervous system? We know the CNS is very energy intensive, so intuitively it makes sense that if the powerhouses are not healthy,
right,
then an energy intensive system struggles. But at this point, how much do we understand in that regard?
Actually, quite a bit, because there are syndromes out there that are mutations in mitochondrial DNA. And so those are hard to recover from because you've lost genetic material that you just can't get back. The other aspect, not only reduced ATP, but the other the side of the coin for defective mitochondria are that they generate a lot of reactive oxygen. So these are things that oxidize proteins and lipids and they can cause damage on their own. So it's not simply the reduced ATP level, but it's also the generation of these reactive oxygens, so it's oxidative damage. And neurons are particularly sensitive to oxidative damage, and that's one of the hallmarks of the late in life neurodegenerative diseases is elevated reactive oxygen. So being able to correct the mitochondrial function as far as the mitochondrial DNA and just overall mitochondrial function will restore ATP, but it's also been shown that it will also reduce aberrant ROS production. So it's a double that you can basically get at both things just by taking care of that one issue.
It's hard to know exactly what restoring mitochondrial function might do, but it's easy to imagine by improving both ATP production and reducing damaging ROS production that there could be significant neural improvements.
Well, you'd hope so. Yeah, you would think that that would be the case. It hasn't been really tried that way because of the way people are doing research now. It's not looked at restoring the mitochondrial function. It's looked at trying to eliminate the ROS levels. So it's not a clear how much the restoration will happen and how quickly it'll happen. It's two issues.
Yep. We have a question from an audience member who has identical twin 11-year-old girls with MED13L. And this person wants to know if there's added benefit to studying twins. Just as a personal aside, we have 11-year-old twin girls as well, and one of them has MED13L. Just a connection with that person who asked that question. So this is really, I think, for all three of you. You know, is there any particular benefit to identical twins in research?
So I'd say, I think if I understood the comments, there are two different types of twins. There are identical twins and then what people call fraternal twins. The identical twins, the utility of studying them is that the girls in this particular case should have the same genetic information and probably share a lot of the same exposures, but there might be differences. And if there are slight differences between the girls, then those are helpful to know that that's not directly genetically related, but could be due to other things, colds, other infections, you know, other issues. And so it helps us to understand what the variability is, even if we keep the genetics constant. If there are twins, and Nick, I don't mean to ask anything personal, so feel free to, you know, just tell me to, you know, hold back. But
Sure.
when we have twins that are very similar, one would say identical, but different at the MED13L, they're a very unique opportunity. We rarely see that. And I don't think those are either sets of twins. But when we see more commonly fraternal twins who are genetically not identical, they actually, as a really, really useful comparison on the other way, because they usually share the same household, the same, you know, sort of other things that are going on, but their genetics at MED13L and usually other genetics are different. So they help us understand, and they're kind of perfect controls for what things would like. There's some questions about, you know, if we were able to fix MED13L, you know, could we get back up to quote unquote normal? And those siblings, those twins sort of help us know what normal would look like because they're exactly the same age and all other things. So for those of you who do find yourself in those situations, if you're generous, and I realize it's double the amount of work, but you provide a lot of insight with your families.
Thank you so much. Wendy, could you expand a little bit on kind of what it takes for someone to register with the Searchlight database and, you know, and how important that is to supporting the research that ultimately can lead to a clinical trial?
Sure. Great question, Nick. So number one, I'll also put a plug in. We right now have sent out something that we call Voice of the Community that's actually very helpful for us to shape the study and shape the clinical trials that we're thinking of. So, and this is agnostic to which company, which technology, anything else. We want to make sure we hear from the community about what are the things most important to you. So if we could come up with a cure, what would you want us to cure? What types of symptoms or problems? And we're trying to then follow your lead in terms of everything. So it doesn't take long, but number one, please do complete the Voice of the Community survey if you're registered for Simon Searchlight. We try and make this, like I said, possible to do from anywhere around the world. So it's online in terms of the registration process. You register, that only takes a few minutes. You upload a copy of your genetic test report, and then you talk to a genetic counselor by telephone. We try and make it easy in terms of different hours of the day, you know, so you don't, if you're working a time perhaps when you're not working, to be able to do this. And then gradually over time, you can do it at your own pace, be able to fill out some measures that tell us how your child is doing. And this is something that we continue to follow as the children grow up. So we actually once a year check in or see how things are changing. Hopefully they're relatively similar, but if they are changing, to see that and have a chance to again check in with that genetic counselor by phone, so that reciprocally, if you have things you want them to know that might not be part of a standard survey, we can help with that. I'll also say that that information is de-identified, so no one knows who you are, but then Riza can get all of those data. So as he's thinking about his next publication, or any other researcher, wherever they are, Australia, Texas, I don't care, but it's a multiplier. So you only have to put your data in once, but it could be 52 different researchers all using the same information, and you don't have to fill it out 52 times. You can just do it one time and then make sure all that information gets out to everyone.
Yeah, I'd just like to say that we're very fortunate that Simons has created this Searchlight registry program and allowed us to participate in it. They've done a terrific job, and we would really encourage all families to consider taking the time to register.
I'll just say one other thing. We're looking very much forward to 2022. So next summer, when we're able to get together, we'll be doing things that are in-person assessments as well. So in addition to what we do online, we'll be doing some in-person assessments, and you'll get a chance to meet researchers from around the world who are really interested in being able to help out you and your family. So look forward to that in 2022.
Great, thank you. We have a participant who has a question about their 5-year-old daughter who has a missense variant. It turns out that one of the parents has the same variant, but the geneticist has created some confusion, it seems, by saying that this is a de novo missense. And I think the upshot is this parent is not getting the answers they need from their geneticist. They've been searching for an answer for 4 or 5 years, and they're really eager to find a resource that can help them make sense of their daughter's mutation, and then how to be most helpful to their daughter. Do you have any recommendations?
So I'll give some suggestions. Riza, feel free to, disagree with me if you like. But so it looks to me like for this particular variant, I think the reason why their geneticist is sort of skeptical is it sounds like the parent and the child both have the same genetic variant. And usually what we say in genetics is, it's sort of, if we see that same variant, we expect to see a similar manifestation. And it looks like in this particular case, the manifestations aren't the same. The child has some symptoms, the parent doesn't. So in that particular case, I actually would say go back to the drawing board. So what I mean by that is I'm not sure how the testing was done, whether it was a panel of genes and MED13L was one of those genes on the panel, or if it was the entirety of what we call an exome or a genome. But if it hasn't been done, I would suggest either exome or genome as a trio, so mom, dad, and the child. And actually, even if it was done, it needs to be looked at again to see if something was missed, because it doesn't feel to me like MED13L is the answer, but based on what it sounds like the symptoms are, I bet there is something there. Mosaicism is technically a possibility, but my guess is that's not the answer unless the test report specifically called out mosaicism in the mother or in the parent. So, and again, if your geneticist can go back and start from scratch and do what I suggested, that's great. I'll just say on a personal note, if this person is in the United States and you need help, just reach out to me personally. You can Google my name and find my email address and tell me you saw me on the meeting and I'll help out.
Thank you so much.
Sorry, yes, I also just wanted to totally agree with Andy about this. And if the variant is inherited from the completely healthy mother, it's very unlikely to be disease causing. And the matter of mosaicism, it's not the case here in this situation, very likely. So as Mandy suggested, and we also happen to see this condition in this situation often, that we find a variant in a gene which is known to be disease causing, and there is a lot of overlap between the syndromes. Maybe the parents think this fits to MED13L, but there is a lot of overlap with other syndromes. And we find new disease causing variants by re-evaluating the previously done genetic tests. So as Bandy suggested, so this, I think it's the best way to do for their case.
Thank you. We have a participant who would like to know what a cure for MED13L could look like. Clearly, this is speculation, given that we're so early in understanding the biology of the disease, or even what type of platform could be used for a therapeutic. But this person's notes that in the video, the girl who was the patient couldn't see, and then they cured her so that she could see. And in that instance, it was through a gene therapy, the delivery of a missing gene. What could we expect to see for our children in the case of a gene therapy?
Well, I'll take a stab at that. The ability to, like I said earlier, the ability to change the genes in later in life after neuronal development's happened is problematic on lots of levels. It's hard to do that right now, at least with the current technology. What can be done, though, is trying to support, in our case, what we're looking at is mitochondrial dysfunction, is trying to support that issue and try to get mitochondrial function back up. That would maybe allow us to now start being able to develop some strategies to reverse some of the phenotypes associated with MED13L. Some things like facial dysmorphism and things like that are probably not going to be affected because that's a developmental thing that's locked in early on. But other issues, like where we're looking at muscle strength and other types of cognitive scores, may be possible. And one thing that we're looking at, and one thing that would be helpful for the community, is development of a MED13L mouse model, because without that, I think we're going to have, that would give us a leg up on to answer that question, what would happen? Because we can do things in mice. So what would happen if we reintroduce the wild type protein into an animal that has gone, and again, CRISPR, ways of doing CRISPR, we can actually do this, that we can then induce the MED13L mutation and let that phenotype become established, and then get rid of it and correct that, and then see how the mouse responds neurologically. That type of, I think, would give us that answer, whether going in with gene therapy would be a helpful solution.
Thank you. So I think what I'm understanding is that gene therapy, in a sense, is the holy grail. It's very difficult right now in the context of the central nervous system in particular, but the technology is developing, and we don't know exactly what the restoration of phenotype would be. But there is hope that there might be some improvement in the neuronal and muscular function, but there are also other elements of the disease that are structural, like the facial features that would likely not be restored.
Using viral vectors. There are viruses that infect neuronal cells, like measles virus, and so people are using those kinds of vectors to insert the CRISPR reagents that you would need to do this. A couple of questions that's not clear, and this is something an animal model would help us do, is even if we correct the MED13L mutation, we don't know how long it would take for the phenotype to reverse, right? So when would we recover full mitochondrial function? When will neurons start acting normally? That's a question that we just don't know right now. And so that would be one of the important questions in the future, is if we can get this corrected, what's the outcome? Right now, we don't know.
Thank you. Here's another question for Randy. In the MED13L cells that you studied, is there any reason to believe that the mitochondrial dysfunction is progressive and will get worse over time?
That's a real good question, because we would assume that the mutations, when they happened, the mitochondria were all functioning normally. Everything in the embryo was just fine, and then this is over time. And what people have seen in other systems and in a variety of organisms, that when you induce a mitochondrial defect, it takes some time before that is manifested into a loss of function as far as enzymatic activity, ATP production, things like that. It is clearly a progressive type of disease, in that the mitochondria do get worse and worse and worse. The question is, do they come to a point where you have to have a certain level of mitochondrial function, or the cell can't survive? So clearly there's a way that this effect does not continually go to a complete loss of mitochondrial function. So there may be a baseline in which it gets to, and then it stops at that point. We don't quite understand how that works, but in other systems where it's been examined, it does go all the way to a completely dysfunctional mitochondria.
And Reza and Wendy, can you talk about whether or not you've seen in the patient data any suggestion of progressive disease, or is there reason to believe that there might be some stabilization, or is it too early to tell?
Maybe I'll say something, and Reza, we may have to banter back and forth here for a second. So in a good way, to me, this is not a classical progressive condition in terms of deterioration that's consistent and seen repeatedly. I do see sometimes that with epilepsy, if the epilepsy is not controlled, there may be a period of things getting a little bit sort of stagnated, or maybe brief periods of sort of going down, but then being able to come back up. And I think, as we all see with our kids, if they're sick or have something major going on in terms of, being in the hospital with open heart surgery or something like that, then I think those are those little setbacks. But I think they're sort of temporary setbacks, not permanent sort of setbacks.
Yes, I also totally agree. From the literature, and also from a collection we have, like about 50 patients now, and few patients also at the ages of 40 to 50 years, we really don't see a regression, because there are genetic syndromes that clearly show regression over time, but MED13L doesn't look like to be like that. And also, from the mitochondrial point of view, I think MED13L is one of those syndromes that the mitochondrial dysfunction is somehow is there, very likely, and the data looks very nice also from Randy, but I personally don't see a full-blown mitochondrial disorder there. I think mitochondria is one aspect which is probably affected, but it's probably not the whole story there, and I don't think that it's a progressive disorder. There's no sign of regression, no sign of progression, rather some improvement. For example, in the motor development, there are improvement. People also start to learn talking and manage themselves in later ages. So it's unlikely to be a progressive, unless, yeah, some of the patients, as Wendy mentioned, that they have some severe seizures, and if that's not controllable, may have some sequela for the patients. They may get worse because of the seizures, but otherwise doesn't look like to be a progressive disorder.
Thank you. We just have a couple minutes left before we get cut off, and we'll go to the next session. I'd like to flag a question someone has about their daughter who has MED13L with elevated lactate and elevated urine organic acids. Their geneticist didn't think this was related and tested her mitochondrial DNA and nuclear DNA, but nothing came back. Is it possible that these elevated levels are actually caused by the MED13L mutation?
I'll just say these are tricky assays to do, and I'm glad to talk with people offline or they can talk to their geneticist. But sometimes we get what we call artifactual elevations in the lactate if they're not processed correctly. So it's tricky to be able to do that. And in a similar way with the urine organic acids, depending on what the metabolic state is. In other words, what you've eaten, when you've eaten it, how much body stress the body's under at the time, you can get some differences. So I think ultimately it's like everything we do in science. What we'd like to see is that there's something consistent, that if it's collected and the assays are performed under the right circumstances, whether or not there's that consistency in terms of those elevations. Little sort of in the weeds to get into for a forum like this, but anyway, I'd say the reason it looked like your geneticist looked for specifically mitochondrial things and didn't see that, whether it's instead due to the MED13L, I think you should do the testing a few times and see if it's consistent.
Thank you. And then in our last 30 seconds here, Wendy, if someone wants to learn more about when and where the 2022 in-person sessions will be and kind of how to learn more about that.
So the 2022 will be August 4 to 7 in Baltimore, Maryland. That's in the United States. So August 4 to 7 in 2022. We'll be sending out announcements so that people will know where the Simons Foundation is subsidizing in terms of the events. So, all the meeting space for people to meet, the research teams that will be there, all of those things, but you will have to pay your own travel to get there and then the hotel rooms. But we are subsidizing a lot of the infrastructure for the meeting. So we hope we'll be able to see people there. As you're doing this, one of the most important things you can do as a community is being able to get your numbers counted. The last thing I'll say, but as companies are thinking about where they want to invest, I'll be really crass, they're looking for market. They're trying to see how many people out there can benefit from this, how many people they can sell their product to. So stand up and be counted. If there are others on the Facebook page, you need to be in a registry to officially be counted, but it's really important to see the numbers. Otherwise, there are other communities, other conditions, they're all important. So I don't want to say anyone is more important than the other, but if they don't see you, they don't know to work with you.
Thank you so much. We're about to get cut off, I think, here, and we're going to have to go to the next session. So thank you, everybody, for participating. Thank you, researchers, for sharing your presentation and your time in the Q&A. Bye, everybody.
Goodbye.